Hello, my name is Soumyaroop Bhattacharya. I'm from the University of Rochester, and today I'm going to talk about our results of single-cell profiling on newborn human lung cells. So the lung is a complex organ. It comprises over 40 different cell types. We don't have much information about how these cells interact among each other especially during the development. So far, we've performed molecular analysis like transcriptomics and proteomics from the dissociated cells of these newborn lungs. However, those have mostly been focused on the bulk cell level. So the goal of this project was to characterize the genetic heterogeneity and cellular lineages of newborn human lung and looking at single cell transcriptomic level. In order to do that, we sequenced the two lung lobes from two newborn human subjects, and the cells were digested and frozen, and later on they were rapidly thawed, and from each of the subjects were used for two single cell captures. So altogether there were four captures. The libraries were prepared and sequenced in HiTSeq 2500, and the data was analyzed in Seurat, and the combination of all four runs was performed using CCA. Then the cells were clustered, and markers for each of the clusters were used through top1 for cell type identification. So the major cell types were identified using their known markers. As you can see, the epithelial cells are marked here, mesenchymal cells marked here in blue, endothelial cells in red, and immune cells in pink. And even though there are more mesenchymal and endothelial cells, but we were surprised by finding some immune cells at that early stage. We further used the markers of each cluster on top one to look at the cellular subtypes, like the mesenchymal cells were identified into further subtypes of myofibroblast, matrix fibroblast. Epithelial cells were identified into their subtypes, and so were the immune cells as well. So we could see multiple distinct populations of each of the cells. So the current information we have on the lung development is primarily derived from rodent models. And even though the stages of lung development are conserved across species, there are differences, major being that birth in mouse takes place in saccular stage while that in human is in alveolar stage. So we used longitudinal single cell sequence data from mouse that was collected at 1 day, 3 day, 7 day and 10 day post birth, and integrated it with our two newborn human single cell subjects. For that, we used 14,500 genes that were matched through Biomart. So in all, we had almost 5,500 humans and close to 33,000 mouse cells. We integrated them using CCA and clustered, and each of the cluster markers were used for cell type identification. So similar to the human cells, the integrated data also had those four major cell types, identified the C's known markers. However, we could see that because the human cells had very few epithelial cells to begin with, we did not see, like, if we could only get less than 5% of epithelial cells. However, we could get at least 25% of all mesenchymal cells were from the humans, and almost 12% of the endothelial cells were from humans. So the human cells proportions did vary by cell types, and we further looked at the cluster markers to identify the cellular subtypes of each of these major cells, and the cellular subtypes were also conserved same as then were almost similar as they were in the humans only. So we used the integrated data set and pulled out each of the cell types separately and to estimate the maturation state of human cells in terms of their age in the mouse lungs. We used PCA within each cell type and identified a PC that was most associated with the age in mouse cells. And then within that PC, which we call PC age space, we looked for 100 all the 100 closest mouse cells for every human cell averaged the ages of those 100 cells, adjusted for the proportion of cells, and then estimated the age of each of those human cells. When we did that, we could see that, as expected, because most the human lung is similar to the mouse lung, which is 6 to 8 day old, we could see most of the cell types were in that range. They were different by a day or two, but mostly within 5 to 9 day range. However, when we looked at the matrix fibroblasts, we could see some of them were in that range, but majority of them were much younger. And one of the markers of these cells was HES1, and using the immunohistochemistry, we could clearly identify cells that were distinctive in terms of the HES1 expression. So to summarize, we have successfully performed a single cell sequencing from newborn human cells. We could get only a few epithelial cells and surprisingly some immune cells. However, we got decent amount of mesenchymal and endothelial cells. And when we integrated these newborn human cells with the young mouse cell, we could estimate cell type specific maturity that differed by different cell types, and we could see the two distinct populations of matrix fibroblasts. And I would like to thank all our collaborators who helped us to complete this study. Thank you.